I also wish to thank the organizers of this conference for giving me the chance to present the results of one of the last projects of my PhD, which I did with Arne at Stockholm University. And the whole thing is about large-scale protein structure prediction enabled by improved contact predictions and model quality assessment. So just to give you an outline on the topic, it's template-free protein structure prediction, which means that given an input amino acid sequence, and you want to know the structure of it. But the problem is that if you search in PDB, you don't find any homologous proteins. So what can you do then? And what you can do is to first start and predict contacts between residues in that sequence. This results in a contact map, which you can plot like this. So what we have here is the sequence position against itself, and you make a dot whenever you predict a contact at that position, or between these two positions or not. And then you can compare it to the native contacts, which we define as being the C-beta distance of below 8 angstrom. And then you can use these predicted contacts to fold the protein, so you can use them as restraints in protein structure prediction. And in this project, we wanted to do that for all PFAM families in the PFAM database. So we want to look at how many PFAM families can we predict structures for, where there is no known structure. And PFAM contains about 16,000 domain families, and it covers more than 45% of the human proteome and over 70% of E. coli. Each of these families is given as an alignment of its members' sequences. And when you take this alignment and search in PDB, you get a hit for a bit more than half of these families, which means that for a bit less than half of them, you don't find anything in PDB. So this is where we want to apply our method and see how many of those can we predict. And the problem is that, so if you look closer at these alignments, the families that have a known structure tend to be much larger, so they have more sequences in the alignment. And we don't measure this in just counting the number of sequences in the alignment, but rather taking into account the similarity between the sequences, and this is called effective sequences. It's a bit more robust. And the families that don't have a known structure tend to be much smaller. And so what we want to do is to predict structures for all of them, and then afterwards look at the models and see which of them are good and which are bad. Or if we can pick up some families that have a good model. Yeah, so this is how we did it. That's the overall pipeline, and now in this presentation I'm going to go step by step through that pipeline and explain you every step that we did. So the first step is to use alignments. So this is what it's all based on. So contact prediction is based on alignments. I'm going to explain it shortly. And what we did here was to use two different alignment methods, and also each of them with two different e-value cutoffs. Because the problem is that before you know the results, you don't know which alignment you should use or which might produce the best result. And this is shown here. So if you take, if you predict models this is just a small benchmark data set, for using just a single alignment compared to models from all 4 alignments, you can see that when you use all 4 alignments, you get a higher model quality. So there, it helps a bit. And we measure this model quality, or I will use throughout this presentation, I will use TM score to measure model quality, which is the similarity to the native structure. And this score ranges from 0 to 1, where 1 means a perfect match, and everything below 0.2 basically is just a random match between the two structures. And if you're above 0.5, it's very likely that the fold of the model is the same as the native structure. Yes, this brings me to contact prediction. So how did we do that part? And as probably many of you know, that given that you can see, or that you so when you're in a protein structure, when you have two residues that are close to each other, you see correlated patterns in the alignment. And you can calculate something like mutual information to get these correlations. But the problem is that you have lots of transitive effects, because here in this case, the gray residue is only close to the green one, but not to the blue one. But the blue residue is close to the green one as well. So they have a correlated pattern in the alignment, and therefore also you see correlations between the gray and the blue one. So this leads to many false positives. And a few years back, people came up with the idea of using a global statistical model on this alignment so whenever you predict the contact between two columns, you not only look at these columns individually, but you look at the whole alignment at the same time, basically. And this approach is called direct coupling analysis, and it is able to get rid of most of these transitive false positives. And we then thought, how can we further improve upon this with contact prediction? And the thing is that if you look at these contact maps, you clearly see that these contacts occur in patterns of interactions between secondary structural elements. So in this helical protein here, you see that the pattern between these two helices looks like this, and a pattern between two parallel helices looks like that. And it's similar, or well, it looks different for beta sheets, but it also produces clear patterns, and they're much more dense. So that's because every consecutive residue is in contact. And you can actually detect these patterns when you do treat contacts during prediction, not individually, but if you look at a surrounding window around each contact. And this is exactly what we did. So we take the multiple sequence alignment as input, and then run direct coupling analysis, which gives us some initial contact maps. Based on these contact maps, we iteratively improve them. So we start for each contact that we predict. We also take the contacts in this window into consideration, and then predict this contact map. In the next step, we use this as input and predict the next step, and so on. And we also use some additional input features from secondary structure and solvent accessibility, and an additional contact prediction method, which is not based on direct coupling analysis. And you can see here why we use this method. So this one is the one which is not based on direct coupling analysis, and this one are just plain direct coupling analysis. And you see that the performance, which is measured in precision, so the percentage of correctly predicted contacts, strongly depends on the family size for direct coupling analysis. But there are methods like this one that are less dependent on the family size. And if we include this method in Pcon C3, which is our contact prediction method, we are able to predict more accurate contacts for small families. And as I've shown before, this is where you want to go. So you want to predict accurate contacts for small families, because most of the interesting PFAM families are actually small. And if we compare it to, so we perform more or less the same as other state-of-the-art methods. And you can use this contact prediction method at c3.pcons.net. Yes, so let's go to structure prediction. So now we have contact maps for these families, and we want to model the structure based on these contact maps. Before, what we've been using was Rosetta, which is based on Markov chain Monte Carlo simulation. And the problem there is that it's just too slow to apply it in such a large scale. So it just takes too long, and we don't have the computational resources to apply it to all PFAM families. But luckily, there's a method called confold, which wraps around the crystallography and NMR system, CNS, and makes it really easy and nice to use with contacts, and it uses a distance geometry approach to basically fit the atoms of the protein against the restraints from the contact map, and it's much, much faster. So in this case, it's about 100 times faster than Rosetta, and the difference in TM score is not that much, so that's why we decided to use confold for this study. And now let me get to the most important part of this approach, is to how do we assess the quality. So now we have models. For every family, we have a pool of models, and we want to rank them. And we also want to say how good is the best of these models if we don't know the answer. So we want to have some reliability score on this. And as I said before, you could do that looking at effective sequences. So the size of the alignment, so the quality of the contact map is somehow dependent on the size of the alignment. But the correlation is even less if you then look at the quality of the structure. So you can do that. You could have a cut of, say, you use all the everything which is larger than 1,000 effective sequences, but it's not such a good measure, so it doesn't really give you a good idea of if your model is really good or bad. And if I say a model is good or bad, I mean that it has a TM score above 0.5 or not. So it's kind of transforming it into a classification problem. And but luckily, there are methods that can look at the actual model, not just the alignment, but then look at the resulting model and predict a score for this model. These tools are called model quality assessment programs, and they basically try to predict this TM score. So they assess the quality by directly looking at the models. And then you can really use a cutoff on that score that you get from this method and allow for a certain false positive rate, for example, and say, okay, at this cutoff, we have this false positive rate, and we get these many models to be accurate, or we get so many models predicted to be accurate. And this is what we did. So we not only used one of these tools, but we used a few of them, and I will just briefly go through them. So PCONS, what it does is it compares all the models that you have for each family against each other, and then its score is based on the similarity between them. So if they all agree, so if they're all very similar, it's likely that they are correct. Whereas in contrast, something like ProQ3D does single model quality assessment, where it looks at each model individually and decides if this model is accurate or not, or it gives it a score by only looking at one model at a time. This CNS contact score is the output from the structure prediction. So where, or it's how they are internally ranked, which means it's the energy that you get from how much does the model agree with the input contacts. So it's basically like the precision of the, or it correlates very well with the precision of the predicted contacts against the contacts in the predicted structure. And then we also, for this project, we also developed just a linear combination of these three, because we could see that we get even more families when using a combination of these methods. So yeah, as you can see here, this is the false positive rate against the number of families that we say are accurate and are actually accurate. So the higher, the better. If you only use the number of effective sequences, you don't get much out of it. If you then include information of the contact map, so if you look at just the predicted score of these contacts and use this, you get a bit more. But if you, as soon as you start looking at the actual model that you predicted, you get many more. You're able to predict many more structures. And okay, so now I've been talking about the ones with a known structure, but how does it look like for families without a known structure? And this is what we get. So each of these tools predicts some families, around 200, and they are not overlapping completely. So some tools predict some families to be accurate, and other tools pick up other families. So if we pool them all together, we get over 550 families to be predicted to be accurate at 10% false positive rate. And there have been similar studies from the Baker lab where they used a different approach and Rosetta to fold the structures. And they, the nice thing is that these two studies are not, they're quite complementary. So since we use this very different approach, we get also different families with an accurate, or with a predicted to be accurate model, and the overlap is not that big. So this adds even more families where we have some, or getting some idea. And if we look at this overlap, over 74% of these have a TM score higher than 0.5, and these are just two nice examples that I wanted to show where our structures really also look like the ones that you get from Rosetta. So yeah, and I wanted to show you, yes, exactly, one example. This is a PFAM domain. So in PFAM, there are many domains that it just says domain of unknown function, so you have no idea what it does, and it's not belonging to any clan or anything. And some of them ended up being in those where we say we have an accurate model. If we then use this model, so the alignment had only 413 effective sequences. It has a length of 178 residues. And if we then use this model and search in PDB, we get a match to a structure, which is or it matches a subdomain of a structure of this 4-thioridine synthetase. And if we then look at this structure, we see that it actually matches a part from another PFAM domain, this PF02568. And this could indicate that this domain of unknown function either could belong to that family or maybe belonging to the same clan of this family or something. So it helps to, at least it gives some hints of what this domain could do. Yes, and with that I would like to conclude. So the take-home message is that we are able to predict contacts also for smaller families, and most importantly that if we use model quality assessment, this is really the key to identifying good models. And then these models can be used to search PDB. And in another example, which I didn't show, they were also able to provide some helpful information. Yes, I should thank all of you.
What fraction of contacts you need to be able to reconstruct the structure?
That's a good question. So we have played around with that a lot and tried very different things. So basing it on, for example, the score of these contacts or just the number. And what ended up on average being the best is taking a cutoff based on the length of the sequence. And in this case, we've been using 2.5 times the length contacts or top ranked contacts. Does this answer your question?
Do you have an idea of how many of the PFAMs you're able to make predictions on are actually in PFAM clans? We did a study a few years ago that we never published that most of the PFAM clan relationships are correct. Occasionally there are inserted domains in one clan member versus another one and pieces of unrelated structure. But for the most part, those remote relationships are correct, and that can help you verify whether your structure predictions are correct.
Yes. We have not looked at it on a clan level yet, but that's definitely something we should do. Yeah, thank you.